Hello everyone. So today I'm going to be guiding you through how we can use two different pieces of TotalLab software to be able to actually monitor our host cell protein content throughout maybe different stages of development, across different sites, production versus R&D, or even different sites worldwide when we're wanting to do some tech transfer, move a process from A to B, or really just get a bird's eye view of how the concentration, the relative quantity of host cell proteins are changing throughout the process from upstream, downstream, through all of your purification, chromatography steps, things like that. So we've made this really, really easy within the SpotMapMS and the TrendLab software, and even the SpotMap ELISA and TrendLab software, but I'm just going to be showing you with SpotMapMS at the moment. So if you've seen any of my other videos on SpotMapMS, SpotMapMS is LCMS-based analysis software to automate the analysis of samples for known host cell proteins and categorize them according to risk, so to give them that bit extra context for what potential issues those host cell proteins could be responsible for, based on where they've been identified in the literature, when either immunogenic or drug stability effects have been mentioned in the literature. So once we've imported our raw trace files, we get this report which kind of shows us all the host cell proteins that have been found, and as I mentioned, kind of what their impact is, what area they affect, whether it is drug quality, drug stability, or immunogenicity. And the risk profile is obviously down to whatever kind of advanced therapeutic that you're making. If it's a vaccine, you can probably get away with a different risk profile versus an advanced therapeutic that's going to be administered multiple times. So once we've performed all of that work, I'm not going to repeat that in this video, but I do have multiple other videos about the SpotMapMS product, the software, about how it works, guiding you through all the steps of the analysis. And if you are interested in, performing automated host cell protein analysis by LCMS, I would encourage you to check out our website. I'll link it in the description below. Check out those videos to see how much time and how much, of a reduction in interoperator variability SpotMapMS can offer as an analysis software package. However, I'm just going to go through the tracking right now. So imagine I've gone through all of my peptide inspect, and I've validated all of my matches or got rid of the ones that I don't believe are a true match. And I finally ended up at my export stage, which is the summary of my work that I've done and what reflects the host cell proteins that I believe to be in my sample and I'm fairly confident about. So I've got two options at the top here. I've got the ability to perform a CSV export and just kind of dump all this data out in a CSV, the industry standard for science, or I can export these results to what we call TrendLab. Now I'll show you TrendLab in a second, but TrendLab is designed to track trends in your lab, hence the name TrendLab. It's designed to be a longitudinal tracking app with various different quality control metrics built into it as well. And yeah, that's what it does. It's akin to a dashboard, but it can be much broader than that. So it allows you to track all of your host cell proteins over time. So once you've completed your analysis using SpotMapMS, you can then come through and click on Export to TrendLab and get all of this information modeled in TrendLab for this particular experiment. So when we come through to TrendLab, you can see this is quite a sparse example here, but we have all of our data kind of graphed over time, and we have these upper and lower bounds for QC metrics, and we have the ability to actually show for every experiment if our SST was a pass or a fail, if you are performing SSTs. So the model that we've used here is a pre-sample analysis SST and a post-sample analysis SST. So your samples would be like a sandwich between two SSTs. These green dots here, you would expect are a past SST. So all of your SST values have been within their expected ranges, depending on kind of the standards that you're using to calibrate. And then we've got our red circles showing that there was an SST failure. So we can change the way that this, this screen looks. We can add on specific proteins that we are, that we want to consider as, host cell proteins that we want to track over time, that we're, specifically concerned about, perhaps. So if you're having a real hard time getting rid of one host cell protein in particular, it's become really sticky, you can just focus on that, or you can focus on all the different host cell proteins that you might be interested in, and see how those are changing at different stages of your analysis. So we can also add different QC fields for the QC of our experiments. So we can do some of MS1 intensity, and these are all the metrics that we use for saying whether it's a QC pass or a fail. And we can obviously include our SST QC fields and set the upper and lower bounds for those SSTs there as well. As you can see here, we've got our SST proteins here, and we can watch how those are changing in time, and we can control whether we see this in intensity or relative quant. We can see sample QC fields. Again, these are our sample proteins, so what are we running here? And if we had multiple tabs, if we had like a really large screen, we could track loads of these over time. So I'm going to show you a different experiment, a really large scale experiment. So each one of these experiments is actually 7 triplicate trace files. So we've got 21 different trace files, all averaged out to become 7 traces in triplicate, and then all broken down into one experiment. So you can see here how we can condense a huge amount of trace files, of information, of host cell protein content into something that still is easy to read, easy to visualize, and look at across time and across site. Again, we've got the SST summaries at the top as well, so we can quickly visually check that our QC metrics for our calibrants, for our calibration, are correct, which means that we can then, trust the results that we're seeing below. So again, the controls are still the same. So we've got intensity or estimated relative quant, and we can turn on or off the different proteins that we're interested in tracking. So turn that one on. I think I already had that one on, actually. So let's grab some of these, pyruvate kinase. So you can see that you can build up the dashboard, the view over time for the particular host cell proteins that you care about. So you can have it one of two ways. You can see every single host cell protein if you really want to be down like that, but if you don't want to see every single host cell protein because it would be too cluttered and hard to identify the things that you need, you can be on the other side of that fence as well, and you can only track the ones that you're specifically concerned about. So if you know in your process that there are particular host cell proteins present, it's an established process, you just want to monitor them, then you can do that for all of your experiments. So you could also have this broken down, these different experiments, into like purification step 1, purification step 2, chromatography step, final release, final batch release, and you should be able to see the estimated relative quant just ideally going down over time to show that your chromatography steps are working, your purification steps are working, and that by the time you get to final release, there's really a completely negligible amount of those cell proteins that you care about within your sample. So yeah, that is the harmony, the hybrid, the combination of SpotMapMS and TrendLab that will allow you to easily visualize your host cell protein pathway, your host cell protein pipeline, what's happening in your experiments, and it can be used between departments to reduce the level of communication required, reduce the number of reports going back and forth, but it also gives you a longitudinal bird's eye view over many experiments as to how your host cell proteins are changing between different batches, different processing methods, and you can easily identify, failures or changes that need to be made to your process. So, for example, if a manufacturer has changed it's a different batch of filtration media or something, for example, you would be able to see if that has an effect on your process extremely quickly and be able to identify that and say, on this date, from this date forward, we suddenly saw a change in our host cell proteins. Can you explain this, Mr. or Mrs. Manufacturer of the filtration or chromatography media, something like that? So it's a really powerful tool to be able to track host cell proteins through your workflow. Yeah, and we're excited to have it. We're excited to kind of push it out into the host cell protein analysis space because there's not really anything like that there at the moment, and I think it's really going to empower people to be able to track these troublesome proteins responsibly in a 21 CFR GMP compliant environment, and really get a much better bird's eye joined up view of host cell proteins throughout all the different experiments and throughout all the different workflows. But if you'd be interested in trying out either the SpotMapMS software or SpotMapMS and TrendLab, please check out the link in the description below, come to our website, leave a comment, get in touch with us, and we can easily set up a demo and kind of get a trial copy into your hands.